In 2010, a small team of scientists at the Max Planck Institute for Evolutionary Anthropology in Leipzig published a paper that quietly rewrote the human story. They had sequenced the genome of a Neanderthal, the first time it had ever been done, from bone fragments recovered in a Croatian cave called Vindija. The sequencing itself was a technical achievement so difficult that most geneticists had considered it impossible only a decade earlier. But the finding buried inside the data was something else entirely. When the researchers compared the Neanderthal genome to the genomes of modern humans from different parts of the world, they found something that the field had debated for years and that many scientists had confidently dismissed. Neanderthal DNA was present in living people, not as a curiosity, not as a statistical whisper at the edge of detection, as a measurable, functional, meaningful component of the modern human genome. In every person alive today whose ancestry traces outside of sub-Saharan Africa, the Neanderthals did not simply disappear 40,000 years ago. They became us, and the consequences of that merger are still playing out in our bodies right now. The image of the Neanderthal that the 20th century constructed was remarkably consistent. Stooped, heavy-browed, crude, a dead end. A species that modern humans, smarter, more adaptable, more socially complex, had outcompeted and replaced. This image was not entirely invented. Neanderthals did have larger brow ridges, shorter limbs adapted to cold climates, and a skeletal robustness that made early anatomists reach for words like primitive. The first Neanderthal skeleton formally described by science, found in Germany's Neander Valley in 1856, was interpreted by some researchers as a pathological modern human, a diseased individual whose bones had been distorted by illness. The idea that this could be a different kind of human, one that had lived successfully across Europe and Western Asia for over 300,000 years, took decades to fully accept. And the idea that this different kind of human had interbred with our ancestors, that the line between them and us was not a wall but a membrane, was considered fringe speculation well into the 2000s. The 2010 genome paper ended that debate. What replaced it was a set of questions far more interesting than the one it answered. The mechanics of what happened are now reasonably well understood, though the details continue to be refined. Modern humans, Homo sapiens, evolved in Africa and began moving into Eurasia in significant numbers, roughly 60,000 to 70,000 years ago. Neanderthals had been living in Europe and Western Asia for hundreds of thousands of years before that. When the two populations met, they interbred. Not once, not in a single location, but in multiple episodes over tens of thousands of years, in different geographic settings. The Near East appears to have been an early and significant contact zone. Europe was another. Western Asia, another still. Each encounter left a genetic trace. The cumulative result is that people of non-African ancestry today carry somewhere between 1 and 4% Neanderthal DNA in their genomes, with the highest concentrations found in East Asian populations, a distribution that itself tells a story about which populations had the most sustained contact with Neanderthals before those populations' ancestors diverged. What is perhaps most surprising is not that the interbreeding happened. Given that modern humans and Neanderthals coexisted in the same landscapes for tens of thousands of years, some degree of interbreeding is almost statistically inevitable. What is surprising is what the Neanderthal DNA is doing in modern human genomes today. It is not inert, it is not vestigial, it is functional, and in many cases, it is beneficial. A 2016 study in Science, led by researchers at the University of Washington, identified specific Neanderthal-derived genetic variants in modern human populations that are associated with enhanced immune function. Toll-like receptors, proteins that serve as a first-line defense against pathogens, in many modern human populations carry Neanderthal versions that appear to have provided significant advantages in fighting the diseases of Eurasia. when the ancestors of modern Europeans and Asians arrived in landscapes that Neanderthals had inhabited for hundreds of thousands of years. Those Neanderthals carried immune adaptations finely tuned to the local pathogen environment. Interbreeding transferred some of those adaptations. People who carried Neanderthal immune variants survived Eurasian diseases at higher rates. Their descendants are alive today. Their Neanderthal DNA is the reason. The same logic applies to other Neanderthal-derived traits that have been identified in modern human genomes. Variants associated with keratin production, the protein that forms hair and skin, appear at elevated frequencies in East Asian populations and have been linked to cold climate adaptation. Neanderthals had lived through multiple ice ages in Eurasia. They had evolved physiological responses to cold that modern humans, arriving from Africa, did not yet possess. Interbreeding accelerated the transfer of those responses by tens of thousands of years. The Neanderthal legacy is not a contamination of the modern human genome. It is, in significant part, the reason modern humans were able to colonize the cold climates of Eurasia so successfully. But the Neanderthal DNA that survived natural selection is only part of the story. What is equally revealing is the Neanderthal DNA that did not survive. A 2016 analysis published in Science by researchers at Harvard Medical School, led by David Reich, mapped the distribution of Neanderthal ancestry across the modern human genome with high resolution and found something striking. Neanderthal DNA is not evenly distributed. It clusters in some regions of the genome and is almost entirely absent from others. The regions where Neanderthal ancestry is depleted are not random. They include regions associated with male fertility, specifically the X chromosome and genes involved in sperm production. This pattern, known in genetics as hybrid incompatibility, suggests that male offspring of Neanderthal modern human pairings may have had reduced fertility. The interbreeding was real and extensive, but it faced biological constraints. The two lineages had been evolving separately for somewhere between 550,000 and 750,000 years, enough time to diverge in ways that made full genetic integration difficult. What survived in modern human genomes is the Neanderthal DNA that natural selection kept. What disappeared is the Neanderthal DNA that selection removed. The modern human genome is not a random sample of Neanderthal ancestry. It is a curated one. Svante Pääbo, the Swedish geneticist who led the team that sequenced the first Neanderthal genome, was awarded the Nobel Prize in Physiology or Medicine in 2022, specifically for this body of work, the development of ancient DNA sequencing techniques and their application to the study of extinct human relatives. The Nobel Committee's citation described his findings as revealing something profound about what it means to be human. That phrase is worth pausing on, because the finding that Neanderthals contributed meaningfully to the modern human genome does not merely change the story of Neanderthal extinction. It changes the definition of what a species boundary means when applied to humans. The classic biological species concept, two populations constitute separate species if they cannot produce fertile offspring, applies awkwardly here. Neanderthals and modern humans clearly could produce offspring. Some of those offspring were clearly fertile because Neanderthal DNA persisted across generations. The boundary between the two populations was real but permeable, more like a border than a wall. What this means for the Neanderthals themselves is a question that science is only beginning to take seriously. The cognitive and cultural capacity of Neanderthals has been systematically underestimated for most of the history of paleoanthropology. Cave sites in Spain, Portugal, and France have yielded evidence of Neanderthal symbolic behavior, pigment use, perforated shells, abstract markings on cave walls, that predates the arrival of modern humans in Europe. A 2018 study in Science, led by researchers from the Max Planck Institute and several European universities, uranium-thorium dated cave art in 3 Spanish sites to at least 65,000 years ago. more than 20,000 years before modern humans reached Europe. The art was made by Neanderthals. Whether this represents something we would recognize as symbolic thought, as aesthetic sense, as the beginning of what we call culture, is debated. But the debate is no longer about whether Neanderthals were capable of it. It is about what it means that they were. A Neanderthal who lived 70,000 years ago in what is now southern France or northern Iraq was not a failed prototype for a modern human. It was a member of a species that had survived for over 300,000 years across an enormous geographic range, that had adapted to climates from the Mediterranean to Siberia, that had developed tools, used fire, buried its dead, and, the genetics now tell us, recognized something in the modern humans it encountered that made interbreeding not just possible, but apparently common. What drove Neanderthals to extinction remains genuinely unclear. Climate change played a role. The climate of Europe fluctuated dramatically during the period of Neanderthal decline, roughly 45,000 to 40,000 years ago. Competition with modern humans for resources played a role. Disease introduced by modern humans, to which Neanderthals lacked immunity, may have played a role. What the genetics make clear is that extinction was not total. The Neanderthals did not vanish. They merged. Imperfectly, partially, with biological friction and significant genetic loss on both sides. But they merged. And the merger is still visible in every non-African person who has ever taken a genetic ancestry test and found, buried in the fine print of their results, a percentage, a small number, a ghost in the genome that is, in fact, an ancestor. The bones in the Neander Valley, found in 1856 and initially dismissed as a diseased modern human, are now understood to represent one of our closest evolutionary relatives. A population that shaped the immune systems, the cold weather adaptations, and possibly the cognitive toolkit of the people who eventually covered the Earth. The story that the 20th century told about them was a story about failure and replacement. The story the DNA is telling is different. It is a story about contact and exchange, and the particular kind of survival that does not require a population to persist intact, only to leave something of itself behind in the bodies of those who came after. That something is measurable. It is in the keratin genes of East Asians that help protect against cold. It is in the toll-like receptor variants of Europeans that have been fighting off specific bacterial infections for 40,000 years. It is in the immune profiles of populations across Eurasia, whose ancestors, on a night somewhere in the ancient Near East or the Balkans, or the river valleys of Central Asia, crossed a boundary that science spent most of the 20th century insisting could not have been crossed. The Neanderthals were never what we thought, because what we thought was a story about endings. What the genome is telling us is a story about what never entirely ends.